Welcome to the SNN Network Virtual Conference. I'm your host, Robert Kraft, and I'm really pleased to bring to you our next panel for the event. This panel is titled COVID-19, Looking at Potential Diagnostics Solutions. I'm very excited for everybody to listen to this panel because we have a great moderator and some very well-renowned guests to join us for this one. But first, I'd like to introduce our moderator. It is John Vandermosten. He is the Senior Biotech Analyst at Zacks Small Cap Research. I've interviewed John many times, and he knows a thing or two about what's going on out there. So with that, John, please take it away, and thank you all for joining us, and enjoy.
All right. Thank you, Bobby. Well, welcome everyone to the panel, and we're going to cover, as Bobby said, coronavirus diagnostics and testing. The subject's been in the news quite a bit over the last approximately 6 months, and it's really critical to understand how it works because we have almost 20 million cases and almost 700,000 deaths around the world. So this is pretty important to what we need to do, an essential component of what we need to do to be successful going forward. So far, geographies that have been able to effectively utilize testing and containment have been able to limit the impact of coronavirus in their areas, reducing the economic, healthcare, and also the death impact of the disease as well. In areas that have not been able to do that, things have not gone so well. Today, we have 4 CEOs whose companies are all developing a diagnostic test for the coronavirus, among other things. We're going to have them share their views on the environment, explain what tests are available, and also show how they work. We're also going to look at the regulatory aspect of marketing a test and the implications of emergency use authorization. There's been substantial criticism surrounding the availability of tests and turnaround time, which is a really important component of contact tracing. And we'll also discuss what some of the bottlenecks are as we face getting enough tests and obtaining results. I think our audience would also like to hear a short summary of what each of the companies are working on. Actually, I'm really excited to hear some more detail about that, and also how it's differentiated from what else is out there. Let's move on to introductions. Bobby already mentioned my name, John Vandermosten. I'm the biotech analyst at Zacks and cover small cap names in the life sciences. And I just want to go down the line. Elliot, Michael, Jack, and James, please give us a short introduction about what each of you do.
Hello, and delighted to be here on the panel. Thank you for the invitation, Bobby. I'm Elliot Goldstein, President and CEO of ProMIS Neurosciences. ProMIS was founded about 5 years ago based on a proprietary technology platform that has multiple applications to identifying specific targets on complex, sometimes misfolded, but complex proteins. So we'll tell you more about that during the panel. Thanks.
Next, my name is Jack Regan. I'm the founder and CEO of Lexigene. Lexigene is developing a point-of-care molecular diagnostic, obviously for COVID-19 and other applications.
Yeah, this is Michael Poirier. I'm the president and CEO of Qualigen out here in Southern California. We've been around since 1996. We are a cancer and infectious disease specialist biotech company. We have a therapeutics pipeline of drugs, drug candidates for cancer and infectious disease. One of our cancer drug candidates we're working on for COVID as well. And we also have a diagnostic platform that's been in the market since 2001. It's a rapid immunoassay system, and we're putting a COVID antibody test on it very shortly. Talk about that later.
Great, and I'm Jim Howard-Tripp, the CEO of State Zero Life Sciences. We're Richmond, Virginia, and Toronto-based. We're a molecular diagnostics company focused into the cancer area. The main focus is really developing a pan-cancer test off of a single sample of blood for early detection of cancer, high sensitivity, high specificity. But most of it's PCR-based, which actually has allowed us to move into the COVID space. And that's where we're currently very busy.
All right. Thank you for the introductions. And let's get started with some basics. I think we hear about a lot of tests out there, but we may get a little bit confused on what they are. We've heard about PCR, IgG, IgM, lateral flow devices, tests that need to go into the reference labs, other ones that are point of care. Can you can the panelists help us understand, what the big groupings of it are? There's two big classes. There's the ones for the active virus and one for the antibodies, but there's all those other items that I also mentioned. Michael, why don't you get us kicked off and kind of explain to the listeners what these big categories are and how they all fit together?
Sure. I mean, when somebody gets infected with a virus, there's different stages that you go through with the infection. The first one is the active stage, where the virus is growing and spreading in your body. And then as your immune system kicks in, it starts to make a variety of different antibodies to fight it and eventually resolve it, you hope. So now you want to test for different aspects of where the patient is in the disease. The first stage being the virus, you'd want to have tests for direct detection of the virus. And so that's where you would look at things like PCR, which are molecular-based tests, which are highly sensitive when detecting viruses. Then as you move through the infection and the antibodies start to be present, what you want to see there is you want to see what types of antibodies pop up. And you have IgM, which pops up first. It's a sign of acute infection. So it's usually present in the active infection stage. So you want to test for IgM, and that test would only be good for maybe a couple of weeks, and then IgM kind of goes away, and then all of a sudden IgG starts to ramp up, and IgG is a separate antibody that will be around for much longer, months, in some cases even years, and that's what indicates a person has long-term immunity. And so that's why you have these different classes of tests, and they're all important. And then within those classes, you have different ways of doing the same types of tests.
Anyone want to take a look at or discuss what lateral flow devices are and how those fit in?
Yeah, I don't mind. We have one, and I think the big thing with lateral flow, when they first came out, they were touted as being point of care. So everyone thought that, what we'd do is send them to the patient's home, the patient would prick a finger and actually use them. And FDA, by and large, it certainly at this juncture anyway, is very much against that. And it's as much to do with, I think, accuracy of the entire procedure, as well as it has to do with the reporting. You certainly can't, it's an infectious disease, it kills people. Authorities need to know who has it and where it is, and you can't rely on patients to report. So I think the lateral flow devices have been very useful until literally a very short while ago. You couldn't do IgM on any of the large autoanalyzers. So the only way you could get IgM together with IgG was the lateral flow devices. And so they proved to be very accurate. But if you look at what we do, because the point prevalence of the disease is only about 5%, your positive predictive value off of a lateral flow device, even one that was extremely accurate, was only about 50% at best. So you needed to be able to reflex to a second test. And if you got a confirmatory test on, it didn't matter whether it's Roche or Beckman or whatever system, you now had a 99% positive predictive value. So that's what's being used these days to back it up.
And Jim, one thing that we talked about before was that that point prevalence and why you go from very high accuracy, but when you have the low point prevalence of 5%, as you said, you only get a 50% accuracy rate, I guess. And I guess maybe you can explain how we get those numbers. And what I think it is that there's so many people that aren't testing or that are negative, even if it's a very high specificity, then you're still going to have a lot of false well, maybe you can explain it better than I can, if you know what I'm getting at.
Yeah. It's if you get a negative, you're absolutely certain, even with low point prevalence, you're not absolutely certain, but you have a high positive predictive value, 99%. It's when you get to the positive as to whether it's a true positive. Mm-hmm. And so it's purely mathematical. And if you go down to a point prevalence of the disease of, let's say, even about 1%, you've only got a positive predictive value of around 11%. Now, as you move on up and get higher, and we're actually testing in Arizona at the moment, and Arizona on PCR tests are showing 15.4% positive. So, you step up to that, and with any of these tests, your positive predictive value goes up again. Purely a mathematical calculation.
Mhm.
Is the positive a true positive?
I think one of the questions we want to ask too, and you raised it, John, is sort of who do you test and how? And there's different paradigms for that. So pool testing, for example, is very interesting, especially given sort of the context here in the US where we have to really ramp up the number of tests. And as you know, what you do is instead of measuring individual by individual, you group together small numbers of individuals. If all those 4 or 5 individuals are negative, you've just saved, say, out of 5 tests, you've saved 4 tests out of 5. Worst case is if it's positive, the pool test, then you got to go back and test them. It's also a great idea if you like to test a group of people in a similar environment, boys in a frat house, for example, or people who attended a dinner or an event. So there's another one that doesn't get too much mention, but that proved to be very valuable, especially in Massachusetts, if I recall. And it's very interesting, I think, for population dynamics, because that's really hard to gauge, especially as the virus is at the beginning and as it's really raging. And that's testing in sewage. I think that's a very, very interesting approach as well. The point I'm trying to make is it sort of takes a village of different types of test approach and technologies, different populations, and pulling all of that together to get a clear picture. It depends what question you're trying to answer.
What, one other question I wanted to ask was just what is the best test we need for fast turnaround? I mean, what would be the optimal test to use to really make contact tracing effective?
It's got to be very accurate, whatever it is. And apologies, Jack, I think you wanted to jump in. The key thing, a lot of things we're seeing, and so I'm going to go back on Elliot's point a little bit with pool testing, because as we were looking at it, it again depends upon the point prevalence. Pool testing with very low point prevalence makes sense. Pool testing as you get higher point prevalence, in actual fact, costs you more. So you've got to figure out exactly where you position it on this. The sort of fecal testing useful, but it appears in stool not all that frequently. And when I say that, maybe about 60 or 66% at the time. It's the same with blood. We had a look at blood. It's only about 12% of the time you pick it up in blood. So part of one of the issue right now is there are these new things that everyone goes, gosh, it's new and you've got to go chase it on the bandwagon. Your question's right on. What constitutes a really good test?
Jack, did you have anything you wanted to add to that?
Yeah, I was going to add to that. And so, I mean, obviously everybody wants a perfectly accurate test in an instant. I mean, that's the desire to sort of shine a light in somebody's nose and get that perfect answer right away. The reality is this is based on, biochemical reactions or affinity, avidity of antibodies to the antigen. And so there is no perfect test in regards to speed and accuracy. And generally speaking, as we've heard the panel say, the antibody tests, they do serve a great purpose, but they're generally not quite as sensitive as PCR. And the reason for that is amplifying the signal. With an antibody test, you're detecting the antigen, and it's very difficult to actually amplify that signal very much. You can do it a little bit with chemicals conjugated, but it's very limited. In contrast, when you're doing a genetic-based test that uses PCR, that's an exponential amplification reaction, but it takes longer. And so if you want that high sensitivity test, you have to afford the time to do it. Generally speaking, for the most part, PCR tests are going to take you an hour, whereas the antibody tests, they can be done in 10, 15 minutes, but they're slightly less sensitive.
I mean, where is the bottleneck? I mean, what test has the biggest bottleneck? So let's say we're policymakers and we're trying to pick the best test to go forward with right now. Which test would that be, based on the constraints we face right now in the United States?
Well, I mean, obviously everybody in this panel has different opinions. And I do think the different tests do have an application. So it's not like there's only one test, right? I mean, the advantage of the antibody test is they're low cost to manufacture, don't require any special storage conditions, they're easy to operate, but they do lack the sensitivity. So you have to ask yourself, what is the harm of getting a false negative result, right? And then on the other side of the coin, you have PCR tests, which historically are more expensive, take longer to do, but are more sensitive. So you get that higher accuracy. So there's a balancing point. We need both. And I think, given the fact that right now in the United States and across the world, there is such a shortage of testing that we really need all branches of diagnostics providing anything they can to at least give us a better opportunity to test more people We've already heard the panelists talk about pooling. I mean, pooling is fantastic, but often pooling puts you at greater risk of missing a true positive sample in that pool because you inherently dilute the amount of virus present. Therefore, you need better sensitivity. And so pooling generally is reserved for the PCR-based tests, not the antigen-based tests.
Correct.
Yeah, that makes sense. You know, let me ask a very basic question, but I think probably a lot of people wonder this. How do you test the test to make sure that, you know, how do you verify what the sensitivity and specificity of the test is? Elliot, why don't you start us off?
Well,
You're my upper left.
I think others should start off because we're not working on diagnostic, a COVID-19 diagnostic test for the virus. We're working on serology tests for antibodies, and so we'll talk about that maybe in the second step, neutralizing antibodies in particular, right? It's immunity you want to test for. So maybe I'll let others answer that question as it relates to testing for the virus.
I'm happy to take that. And so the FDA asks you to test your diagnostic against a comparator. In many cases, that's an already approved, an FDA-approved comparator that is PCR-based. And so for us at Lexigene, we have to run our test, and we compare it to data generated at a reference lab by an already FDA-approved RT-PCR chemistry. And I've just introduced the word RT. We haven't talked about that yet. RT is just an acronym that stands for reverse transcription that is required to actually take, an RNA genome and convert it into a DNA template to actually do PCR. My apologies. But anyway, we have to compare ourselves to something that's already through the FDA. And that other result is often considered the gold standard in your comparison. So when you run your diagnostic and you compare it against the ones already through the FDA, you may have a discordant result. You may in fact be right. The comparator that you compared against may be wrong, but the FDA actually doesn't really care so much. Like, this one's already through the FDA. This one has been blessed, so to speak, and you're not quite there yet. And so you really have to compare yourself against something that's already been through the regulatory process of FDA approval.
Could I ask Jack a question? Jack, what would you do in the condition where there is no reference, at least for the technology you're using? Or a different approach that hasn't been referenced, where you're first? That's our case with SPR, for example, right? Surface plasmon resonance, which actually has extremely high sensitivity and specificity, but it's a research tool. But, well, it's changing now. But what's your thought on that? Not on SPR particularly, but on, a test that's new.
Well, I can tell you what I know. At least in my space of RT-PCR tests, there are many predicate devices out there for which we can compare ourselves to. In the absence of that, my understanding is you have to do a de novo test through the FDA, where it's a complete de novo. It requires more work, more validation, and you have to still compare it to something else that's currently being used, but it may be completely orthogonal or whatever, if that's the right word, diagnostic, and that's the best they can do. But then once you're through, you are sort of the gold standard for that type of technology.
Yeah, I can add a little bit about the PCR test. In our case, we didn't develop our own test. We used the Thermo Fisher test. And however, even with that, even though it came in with the EUA, we still had to validate it internally within the lab as if it was an LDT. So that's the additional layer that goes in this. So we had to go to another lab. We went to some of the hospitals close by, and what we did was we actually pulled from their samples and we ran it against that. And we had to show that what we brought in from Thermo Fisher, in actual fact, performed in exactly the same way in our own hands. So there's reasonable controls.
And I think it's important to note that people don't have a good understanding of what it takes to go through the FDA approval process. And actually, the bar has been increasing, getting higher and higher the farther along we go. When the FDA first granted the EUA, you literally, everything was contrived samples. You didn't actually need to test a real clinical sample. You get through very, very quickly. And so those companies that got through very, very early because they were primed and ready to do so had to jump a very small hurdle. Now there's a lot of molecular diagnostics that are through the EUA process. So the bar has increasingly gone higher and higher, and the amount of work they're requiring vendors like Lexigene and some of the other panelist companies has become more difficult. And we have to do now more work to prove that we can enter the market. And it's just because the FDA is, of course, trying to guard against the safety for the patient. Obviously, we can talk about this as well. There are many suppliers who have put tests out in the market which don't work very well. And the FDA has now started to effectively rescind those EUAs and say these tests are no longer recommended for use. So there's been a very interesting dynamic with the FDA trying to meet the demand of the public for tests, not hold things back, but nonetheless still maintain quality. So we're seeing a little bit of a seesaw effect right now.
Yeah, John, you asked the question about bottlenecks and all that. One bottleneck we found, we've been able to get over it because of connections we have, but is the availability of proper qualified sera from patients. And we've been using thousands, if you like, especially as we're looking for specificity tests, right? We have sera with various coronaviruses that are not SARS-2 COVID, for COVID-19. But that's a real problem for a lot of testers, at least those who may not have the sort of the clout of some of the big boys. That's one sort of restriction.
Yeah, and if you're, sorry,
Go ahead.
I was going to say, bottleneck too, if you're actually doing the testing, it's still availability of supply.
Yeah.
And
Sure.
if it's particularly PCR, it's still not easy to get supplies. So, and you look at the bottlenecks, it's equipment, it's everything. I think it was actually the LabCorp CEO that was out this week saying that I think they've ramped up to doing about 11,000 tests a day. And he was facing the criticism that they're waiting 7 to 14 days for people to get the results. And he said, one, it's everything about availability of equipment. They bought every single piece of equipment they can out there. They can't get more. And it's about supplies. So there's a huge aspect to that.
Yeah, I'd read some of the similar things in some articles all throughout the whole country about different supplies that are problematic. We were talking about FDA approval and the process there, emergency use authorization. What kind of accuracy levels does the FDA look for when they're either giving an EUA or ultimate approval for a test? What's the threshold that they require?
So I can speak to that. In regards to RT-PCR, they're looking for a sensitivity of roughly 95% and specificity equally high. And so that basically 95 out of 100 samples you should be getting right.
Okay.
Yeah, for serology tests, it's 90% sensitivity and 95% specificity.
Okay. Okay. And then what else do they look for when they're granting that EUA? And then when that's granted, how do you get to the next step? What else is required to get full approval?
Well, for serology tests, and I don't want to step on anybody's toes here, but for serology tests, they're looking for cross-reactivity, and they're looking for interfering substances. So it's fairly simple compared to like what you would do for a 510K.
Okay.
If you were doing a 510K, you'd probably be doing at least 4 times more studies.
So that's going to take a little bit of time to get that full approval then.
Right, and as was stated earlier, it would be the first one in is going to have to do a de novo, and then everyone else after that would come in with 510Ks. But this EUA is probably going to last for some time. So then, why would somebody bother?
Yeah, right.
By
the time that happens, it'll be over, right? And we'll have a vaccine.
Yeah.
And like at a lot of FDA approvals, even standard approvals for therapeutics, which is something I've been involved in for 30 years, there are post-marketing commitments, and here there's a commitment to actually finish the file. So you cannot just sort of ride on your temporary driver's license forever, right? You can also lose it, of course, as we've seen with hydroxychloroquine. It's a great example, is that if subsequent data show, I'm just using that as as an example that's in the news, but if subsequent data show that your test or your product or whatever it is doesn't have the required safety or performance standards, you can lose that approval. And we've seen that happen, which is appropriate. So you're still, it has to be a real commitment, to finish your file, whatever process is entailed.
And
I can give you some numbers. I'm sorry.
I'm sorry. I was going to add a little bit more to what's required to get FDA approval, at least on our side. And so, as you just mentioned, you have specificity, sensitivity, sort of your LOD, limit of detection. That also includes inclusivity, where they actually want to make sure your test hits multiple different sort of strains of coronavirus that are still within the family of coronavirus, because obviously it's an RNA virus, it does mutate. They want to make sure that you can actually inclusively detect the circulating strains of coronavirus. They're also looking for stability of reagents. So this is both shelf life stability time as well as on instrument stability time. And if you're actually putting an instrument through like we are, it's also software validation, electromagnetic compliance, and just general safety issues with the instrument that need to actually be certified that you're not going to somehow endanger the operator of the instrument.
Michael, did you say you had a few numbers?
Yeah, I've got some numbers if we're interested. So these are numbers of tests that have already been approved under EUA.
Mm-hmm.
So on the molecular side, there's 116. Laboratory developed tests molecular is 37. There's 2 antigen tests, and serology tests, there's 31. Now, also to note is 72 tests have been pulled by the FDA.
Wow. So that means you said, what did you say, 116? So I guess that was a lot higher. I mean,
molecular tests have been approved, and then laboratory developed tests that are molecular based. So these are within laboratory use tests. Homebrew, I guess is what you'd call them. 37 have been approved.
Okay. Well, since we're talking about numbers, another thing I wanted to touch on was the government money that's supposed to be available for this. Obviously the testing is mostly a public good, because when you're testing, you're probably helping out society more than yourself. So that's probably one of the goods that should be supported by government funding. But how easy has it been to access these funds, and how much is available? The Rockefeller report went into some of these numbers. I think they said 25 billion was allocated. I don't know how much of that is available, but how have you all seen ease of access to those funds, and what's the process to get them? And any other comments on that topic as well? Jim, maybe you want to kick us off there.
Start off with that. So we've only looked for some, because we're not developing our own individual tests. We've really looked more for sort of growth of the company, maintenance of the company, and then I'll go to the reimbursement on the cost of the test in a moment. So certainly the money we were trying to get, some of it was very available. So for example, Paycheck Protection Program, as we talk about, that was very, very straightforward to get. However, some of the other money has been impossible to get. For us, what is more important is getting reimbursed on the test. And we find that's just a huge issue. I haven't yet spoken to anyone that says actually it's easy or it's working. And we were talking with a group just last night. They did 10,000 tests in the last month. They say they're over $1 million out of pocket. They haven't had a penny yet. And this is the 3rd month, and they're still trying to do it.
Now we have a few different payers out there. I mean, has there been a difference? There's the insurance companies, there's the Medicare, Medicaid, and the government payers. I mean, is there any difference there? Or
by and large, people seem to have a little more success with government, because at least we know what the rates are. I'm not hearing of anyone that is actually having success with the private payers. It seems to be just an immense problem. And so, as if you look a lot, our testing is cash. It has to be, because I think right in the very beginning, even the very big guys were saying that without getting paid, we just can't keep doing this. And we can't sit out there waiting for 3 months to get paid. So therefore, we just don't do it.
Okay. Anybody else have similar observations?
Well, I can add something. I don't want to sound like I'm complaining, but I'll do a little bit of complaining if you don't mind. So the NIH, set aside $1.5 billion, for diagnostics, and one of their goals was to allow for 6 million tests per day by December. And so when they do things like that, they're specifically targeting sort of high throughput testing, and you can appreciate that. There's a need to do lots of testing every single day. Granted, Lexigene did apply for this grant money, hoping to be allowed. There was effectively, there's 4 categories for this particular grant call. We fit into one of the categories. There were effectively 600 applicants. Only right now, 1 applicant is advanced to phase 2 for that one particular category. And so it's super frustrating, because they're putting a tremendous amount of dollars into 1 or 2 horses in the game, rather than sort of diversifying the money to particularly smaller companies that arguably needed a lot more. There's also been some BARDA grants given to some very large companies. These are companies that are multi-billion-dollar companies that have manufacturing in place and the ability to push tests out very quickly. Fantastic for them, but I would certainly like more dollars going into startup companies which are trying to launch a brand new technology to bring something new to the market that currently doesn't exist. Because it's really the small companies that don't have the resources to push and go faster, whereas bigger companies with millions and millions, if not billions of dollars of revenue every year, can leverage their massive workforce and, for that matter, treasury to get work done regardless of government assistance. Where it's really the smaller companies that need the help more so from the government than the bigger companies.
And Jack, you mentioned BARDA. We haven't applied at all to BARDA, but I have a personal past experience with BARDA. For small companies, it's a bit of a nightmare, just learning the process and just navigating it. It took us 2 years to get a kind of a novel GCSF, pegylated GCSF into the stockpile. For radiation sickness, actually, from neutron bombs, if you can believe it. But anyway, and it's just such a convoluted, bureaucratic, difficult process. We're a Canadian company, although obviously we like to operate globally, but we're Canadian-based and publicly traded. There are opportunities in Canada that are special and fairly easy to navigate, depending on local factors. So the provincial governments do have sources. We're in discussion with several provincial governments to sort of bulk buying of tests, if you like. So a single sort of contract, and we'll cover all these things. We're getting some pretty good traction on that. The province of British Columbia is quite generous. We've also received the digital supercluster grants. I'll say a bit more about that. But those are very specific to Canada. It's a difference. There's a little less competition, if you like, and a very sort of central focus of the government, what they're looking for in about 6 or 7 different categories. So it's quite easy to navigate and determine whether you really qualify or not, which is part of it. There's no point in wasting, tons of project team time to try and, apply for something that you'll never get. So National Research Council of Canada, we found them actually very cooperative. Yeah, a little bureaucratic, of course, but so local conditions make a difference too.
Michael, any thoughts from you on funding or access to research funds or reimbursements?
We were very successful in getting the PPP money to handle payroll, but we have not had any success getting grant money for developing a test.
Okay.
And, what Elliot was saying, years ago, we tried to go through to get some DOD grants for some other things we were working on, and it was virtually impossible.
This is where lobbyists help.
Yeah.
And we can't afford to pay lobbyists.
Yeah, well, that's just it. I mean, it's pay to play, or it's who you know when you're in that arena. And to your point, the big companies that don't really need the money get it because they can afford to grease the right palms.
Right.
Yeah.
Yep.
I'd like to add one more thing on the regulatory side, because this kind of teed me off too. But this whole EUA process is kind of interesting in that the way it works is, you file your application, but you can immediately notify FDA and start selling. And the problem is, though, and I think this is deceptive, a lot of people don't realize this, is when you notify the FDA that you're going to start selling while they're sitting on your application, you're very limited to where you can sell the product. And that comes into the certification under CLIA of where the testing can be done and what certification the user has to have. So all laboratory testing is really into 3 buckets as far as categorization for users. It's highly complex, it's moderately complex, and then there's a wave category, which is the things that you see in over the counter or in some doctor's offices. And if you submit something to the FDA under the EUA and then you want to go ahead and immediately start selling it, you're limited to the highly complex sphere, which is, not that many places really, not decentralized places. Let me put it to you that way. And I think that's part of the issue, is we need to see these tests getting out into the decentralized areas. Now, that wasn't so much of a problem when they first started the program, because there was probably a couple of weeks lag between submission and approval on EUA. But now you're talking months of delay because there's so many applicants that are looking to get their stuff approved.
Right.
Okay, just sort of final comment on this. One of the things that we're seeing is groups like the counties, for example, are actually getting very creative. And so, for example, working with one county right now, they're actually relying on CARES funding. So it's a way, because you can't do it in the usual route, that's another way to do it. But they understand that the only way they're actually going to get the testing is that they've got to pay for it, but they can then look for reimbursement. So instead of us trying to do it, which we would never get, they can. So sometimes there are those routes. And then I think also picking up on the point that you made, that it's very spotty across the country in terms of where you can actually get the tests. And you're absolutely right. In a lot of the major centers, there's quite good availability, but you move just outside of that, and there's very poor availability. And so that just exacerbates the whole situation. So getting that even, getting that spread out, getting that accessible is key. And right now, I mean, we're absolutely inundated. We're in the market with the test, whereas I think some of my colleagues aren't. But we're being absolutely inundated with requests for it. And it goes back to what's the bottleneck? The bottleneck is making sure you've got the supplies. It's a very interesting game. And then you've got to get once you do that, you've got to get paid. It's not working. It's just not consistently working.
Well, that brings us to another public policy question, which is, what kind of testing plan is needed? What does the government need to do to efficiently put in place a structure to effectively test? And then what else is needed? Contact tracing, maybe a few thoughts on what kind of plan we need to put in place to be successful.
Okay, I'll stir the pot. I'll start to begin with. I'll tell you what's not needed: just testing. Just testing actually doesn't work, because you're getting, everyone believes, of course, by doing a test, I'm actually solving the problem. We're working with one company that has multiple locations. They've had 3 outbreaks in 3 weeks in the same location. And we eventually got them to understand they need a plan. You can't just test, you need a plan. You've got to sort it out. You've got to figure out how you're going to do all of this. And the biggest thing that we're seeing out there is that by and large, there's testing, but there isn't a plan.
Okay. Anybody else on the thought about a plan to put in
Well,
place?
I think what you've got to do is you've got to attack it from all sorts of angles. I mean, you've got the people who have symptoms that have to be tested for the virus. And in a lot of cases, what we're hearing is it's very, very difficult to get those tests. You can call your doctor and say, gee, I've had a fever for 3 days, and they may say, well, yeah, come in, we'll test you. Or they may say, you know what, if they don't have the availability. Then on the other side with the antibody tests, it'd be nice if you could test a lot of people who have no symptoms to see if they've already had the virus at some point in the past. So that's where a good serology test would help. I think you just have to attack it from a lot of different angles, though. And to the point that was just made, I think testing is just one piece of it. You've got to be able to take action once you get the results on people. Either they've had it, so okay, that you treat them a certain way, or they haven't had it yet, they have to be treated a different way, or then you've got the active infection.
Certainly. Yeah, I mean, just in terms of quarantining, in terms of contact tracing, what else? What other things could be put into place besides those?
Well, one question. Oh, sorry. Go ahead, Jack. Go ahead.
Well, I was going to venture a crazy idea, if you don't mind. So with proper, I would say, motivation or direction from the government, we have some very powerful pharmaceutical companies, life science companies, and academic labs that certainly could be motivated into motion to help out. We've talked about reference labs not having the ability to source these high throughput pieces of equipment to help scale their testing. A lot of that equipment currently exists, in labs doing other projects. Mm-hmm. And from a public health standpoint, you might argue, listen, sort of the National Defense Act or whatever it is, say you have to stop doing the work you're currently doing. You're going to train your highly skilled people to do this test. Here is the CDC test. Start doing it, even though you're not a certified clinical lab. It's as best as we have right now, and something's better than nothing. And so right now, I would argue, given the trouble we're in with testing, we need to start motivating some of these other private institutions that aren't typically into providing diagnostic testing to temporarily try to provide services, because arguably they have the expertise, they just need the protocols in place. And if given the proper chemistry, which we all know that what that is, you can just use the CDC chemistry, they could get up to speed relatively quickly. And right now but it does require an act of government at the highest level to motivate that to happen. And otherwise, from the academic standpoint, why would they do it? That's not within their normal scope of work. They have other things to be doing, but they could transition their work to help out with this problem.
Yeah, we'd sort of say also it's turnaround time. It's Dr. Fauci is absolutely correct when he says 7 to 14 days to wait for your result. Waste of time doing the test. It's absolutely correct. Your turnaround time's got to be very quick. I always go with the South Korean example, which is 66%, at least they tell us, of their infections came from that single 31-year-old woman, and she caused a spread in literally a 7-day period. If one person can infect 59,000 others in a space of 10 weeks without it being controlled, you can imagine that you actually have the active virus and we're not telling you for 14 days. It's crazy. So that's got to get fixed as well.
I wanted to ask a question about this. It's more about, I don't think, we need absolutely, when you ask what's the plan, the first thing is you have to have a plan. I mean, there isn't even a plan, but even more than that, strategic intent. So the question I would ask is, with again, so much virus circulating in the United States, or at least certain parts of the United States, where would you prioritize the testing? Because it's not going to change overnight. What are the priorities? Is it legitimate? Can you really do contact tracing and isolation? Well, you can do a certain amount of it, but would it be, is there just too much virus around? And do we need more dramatic boundaries and barriers, if you like?
Well, sorry.
Sorry, go ahead, Jack. Go ahead.
I was going to say, like, right now, in regards to contact tracing, I think that's something which is going to come into play as hopefully this first wave starts to peter down and we can start trying to get a hold of it. Right now, there are so many people with symptoms that they need to take priority. Yeah. And we're not even close to being at the point where we should be testing people who are asymptomatic. Although ideally, we're testing asymptomatic people to see if they're either asymptomatic or pre-symptomatic. Right now, as we just heard, there isn't enough testing supplies. We're now starting to pool people who have symptoms. And so until we get through this first phase, and if history is any lesson here, there are some waves of a pandemic where it'll peak and almost disappear, and all of a sudden complacency, returns, people stop socially distancing, and the virus comes back. And so maintaining vigilance, for the long term until we have vaccine and herd immunity is going to be absolutely required for this.
Yeah.
We did quite a bit of work with New York State, and I've got to tell you, just hugely impressed in terms of how they tackled all of this and how they pulled all of the academic institutions together. They pulled them into a single organization. They took the lead pathologist across the largest of the groups. They put him in charge of it. They liaised with the neighboring states in order to get a coordinated effort so that you didn't get people crossing in both directions. And we all know the outcome in New York. It's really, really good. I'll talk to a university we've been working with too, as they're opening up with this. And it's amazing, because it's got to be very quick testing, but they're doing both PCR and antibodies because they want a baseline on where it is. They're putting supplies into the unit. They're doing temperature checking each and every day. They're doing symptom checking each and every day. And they're confining the students to campus. You come in, you stay on campus, and you run. But by having immediate supplies, if someone shows the slightest symptom, they can immediately get them tested. And then it's the whole contact tracing thing. And at least if you look at it, you've got an opportunity to actually open and then remain. You can control this thing. But now take it out into the broad population, that's just not there. So it also becomes more difficult, but it's not there.
Let's take a little bit different turn, and I wanted to explore something else, and I don't know if there's much here, but does testing play a role in developing successful treatments and vaccines? And once we finally get I think we do have a couple treatments that are working, but some better treatments and some good vaccines that are effective. What role does testing play when that comes about?
I'll take a first stab at testing. I'll talk about vaccines. This whole warp speed development, I think it's very clever. You have to compress the timelines. We can't take 3-4 years of clinical testing and get the quality of efficacy and safety information we need to put the vaccine on the market. It's just not possible, right? But with this warp speed development, vaccines are accepted with minimally acceptable data, which is fine, but that means they have temporary driver's license. If you could, they're adolescent at best, sort of in their maturity. And we would be missing several years of efficacy and safety evaluations, right? And many unanswered questions with these vaccines, the quality of the immune response from different vaccines, the Moderna vaccine versus the Oxford, we don't know, they're very different setups. Duration of antibody immunity. Is it 6 weeks? Is it 6 months? Is it beyond that? What about the quality of immune response in subgroups like the elderly or children? And I would just add to that, in addition to the, how serology testing can help us, there's one other aspect too, of course, is that we're working on a plan in the supercluster network, which is focused on AI specialists predicting likely future mutations. And once they do that, we'll apply our technology to identify epitopes there. But so, that's another aspect that to think future oriented already, because we know it's coming, right? Nevertheless, we've got huge problems right now to deal with. I'd also just add one thing, at least my view is I think has to be coupled with that, especially as we're talking about vaccines, intensive outreach and education to increase resistance to vaccination. It's absolutely imperative. If not, it's not quite a waste of time. We won't see the kind of results we want. So that's just sort of my first thoughts on it. It's going to be really important to monitor the vaccines. We're talking about hundreds of millions of vaccines, if you will, vaccinations. We're going to need to monitor those to start getting that longer term data about safety and efficacy and just dose and, boosters and repeat and all that stuff we need to know. So those are my first comments on vaccination in particular.
Okay.
The only thing I would add is in regards to how diagnostics can help with the other two branches, vaccines and therapeutics. When vaccines finally become FDA approved, there's going to be certainly a shortage. It's not like all of a sudden we're going to have 4 or 5 billion doses to give out. There's going to be massive demand for these vaccines. This is where diagnostics, specifically the serology tests, will come in hand, because right now there's at least a significant percentage of people out there who have been exposed and didn't even know it, and they had asymptomatic disease. And so arguably, everybody who wants to get a vaccine should have a serology test first to confirm whether or not they have existing antibodies. If they do, they should not receive the vaccine. It should be going to somebody who's at a high risk of having complications for a coronavirus infection.
Well, it looks like we have only about 10 minutes left, and I wanted to give everyone a chance to talk about what they're working on and how their test is different from what else is out there. And Michael, why don't you start us off?
Sure. Yeah, our test is, we're being, I guess, opportunistic in that we've had our rapid immunoassay system we call FastPak, which is a laboratory in a foil pouch that runs on an analyzer that's the size of a shoebox. So that's been out on the market since 2001 with things like prostate-specific antigen, testosterone, thyroids. We have about 10 tests on the menu. But it's a magnetic particle chemiluminescence system, so it's a highly sensitive rapid system, and to put an IgG test on for SARS-CoV-2 was a fairly simple undertaking, but one we could do with high sensitivity. We haven't started actually shipping yet. That'll probably be in the next few weeks, but that seemed to be a natural fit for us. But our main thrust is therapeutics and cancer specifically. It turns out that one of our cancer drug candidates, which is a synthetic DNA aptamer that targets a protein that's expressed on the surface of cancer cells, also we feel could attack COVID-19, because it appears that the COVID virus uses nucleolin to facilitate entry into cells to take over the cells for replication. And we feel we can block that by treating a patient with AS1411. So we're hoping to be in human trials before the end of the year with that.
Great. Jim?
Yep. So we in the COVID-19 space, it's PCR-based testing as well as the antibody. On the antibody side, as we were talking about earlier, we use one of the lateral flow devices, but we actually also use the Beckman system. We do both of these because they're gold standard. Beckman is actually just introducing their IgM, so they'll be the first of the large analyzer systems actually with IgM, so that's useful. But I think equally as important as we're looking at it is sort of where you go from here. And so we're seeing quite a lot about, for example, we keep hearing about home testing, and it's a misnomer. Home testing does not exist. FDA does not allow it. What FDA does allow is it allows home collection. So when you get that through, you will get that. And I think everyone's still trying to make sure that it's as accurate as, for example, the nasopharyngeal swab done by a healthcare professional. But it's moving from there to saliva testing. It's moving there to a very innovative set of groups, for example, blood spots. It's moving from there to looking at devices that in actual fact paired with your phone. So you would actually mail it to the patient will use it, it'll link with the phone, and the result comes back to the lab where it's actually analyzed. So there's some very, very interesting aspects, and they're not just antibodies, but the antigens as well. So I think where this is going will be interesting. What we actually do for a living, we talk about, we're an mRNA gene expression group, very novel technology. We actually can focus on all diseases. Our main program's called Aristotle, and been in development for about 20 years. We know that we can find diseases early. We know we can see them before you actually get expression of the disease very often. We can find things as diverse as schizophrenia versus Parkinson's versus congestive heart failure versus rheumatoid arthritis versus Chagas disease. And then, of course, you're in the cancer side. And I think, as I mentioned earlier, the major opportunity for us right now, and we should have it in the market by year end, is actually off of a single sample of blood. It'll be 10 discrete cancers. And the way we interrogate the genes is that off of each of these, for example, like colorectal cancer, we'll actually have the first test that does polyps only. We'll also have the first test that would tell you whether if it's a frank lesion, stage 1, stage 2, or stage 3 or 4. So pretty exciting.
Elliot?
Okay, I'll try and be brief here. But PROMIS Neurosciences, as the name implies, PROMIS stands for protein misfolding. Our technology platform is based on 2 proprietary thermodynamic algorithms that run on supercomputers. Basically, what it allows us to do with these proteins, we'll talk just about COVID and the RBD, the receptor binding domain of the spike protein. We've been able to identify 18 what are called conformational epitopes. So for the lay people in the audience, an epitope is a site on a protein. It's a small part of it that an antibody or even a small molecule drug will bind to it. Okay. But what's really special about these targets or these epitopes, if you like, is not just where they are, but their shape, which is the conformation. It allows us to make or detect antibodies that fit extremely tightly with extremely high affinity on the order of 10 to the minus 12 a molar. So what we do with these targets is we can make antibodies against them. That's what we do for neurodegenerative disease. Vaccine, the targets themselves can be warheads on vaccines, and of course diagnostic tests. In terms of our serology test for COVID-19, it's based on the receptor binding domain, and we'll be submitting that for approval very shortly. But more importantly, what we're looking to do is to see, can one or more of the 18 targets loaded onto the SPR chips, we use surface plasma on resonance, often called BioCore, because they were one of the first companies to develop it, to see if actually the antibodies we detect with the serology test are neutralizing. So we just have a 1-2 suite of tests. Third thing we're doing is working in the digital supercluster, as I mentioned briefly before, again, as it relates to COVID, to predict the likely mutations and then apply our technology once again to see what epitopes or conformational epitopes, so these targets, may appear on the mutated forms. And then again, we can apply the technology for diagnostics, potentially antibody therapeutics, and of course, vaccines. So that's what we do in the infectious disease COVID space.
Great. And Jack, I'll make sure Bobby doesn't cut you off.
Okay. So quickly, LexGene, we're a publicly traded company. We've been in business a little bit over 3 years. We anticipate launching our first product at the end of September, obviously for coronavirus testing as well as other infectious diseases. So our value prop is we do PCR, but we do it at the point of care. So we have automated the PCR process into an instrument that is extremely easy to use. Collect the sample, load it, less than a minute, you start PCR and get that result in just over an hour. And so right now, we're in the process of starting manufacturing. We expect to be starting our FDA study in August, completing that in about a month's time, submitting to the FDA. And right now, they are prioritizing genetic-based testing platforms. So we certainly hope that we'll get approval and be on the market for COVID-19 testing in September.
Great. Well, I think we also want to have everybody share where people can find out more information on their companies. I'll start off with Zacks. Please go to scr.zacks.com and you can find all our research there. And each one of you, please slowly, I guess, Elliot, Michael, Jim, and Jack, just slowly state where people can go to find more information about your company.
Sure. Two sort of main sources, our website, so promiseneurosciences.com. It's spelled promise, P-R-O-M-I-S. There's no E, promiseneurosciences.com. There's a lot of information there. As well, we put out a podcast once every, about 3 or 4 a month. And we've done several on, it's called Saving Minds, because that's, of course, our main focus. But that's also an interesting source where it's not an infomercial source at all. It's we have invited guests, key opinion leaders, regulators, health authorities, that sort of thing, and talk about important topics that relate to us, including COVID-19. So Saving Minds podcast and the website.
Great. I will go and look. You'll find us on www.stagezerolifesciences.com.
Michael?
Yeah, the easiest way to find information on us is to go to our website. The easiest way to find that is just to Google Qualigen, Q-U-A-L-I-G-E-N.
Very good.
And it takes you right there. We're also a publicly traded company on NASDAQ. Our ticker symbol is QLGN, so you could punch that in.
Excellent. Jack?
So yeah, company's Lexigene, spelled L-E-X-A-G-E-N-E. So lexigene.com, and that's why we're publicly traded.
Great. Makes it easy. All right. Well, thank you so much for this. I really enjoyed it. I love doing the research and talking to each one of you to come up with these questions that we covered today. And I hope all the listeners and viewers found it educational and learned a little bit about COVID testing and the companies that we had here today. Bobby, you want to have your closing speech and drinks?
Thank you. Jeez.
Where are the drinks?
Looked like I just went through the ringer. Thank you, guys.
Yeah, the virtual refreshments is a real problem, you know. We have our AGM on Wednesday next week. I mean, it's kind of virtual.
Wait, Elliot, real quick. Sorry, I want to hear more about that, but real quick, John, just need your disclosures real quick on whether any of the companies here are current Zacks clients.
Yes, Qualigen is a client of Zacks, and Promise Neurosciences is also a client of Zacks.